Hi guys, welcome back to my channel, The Lab Code Girl. And today we are going to explore the NCBI and EMBL websites, and I'll be guiding you through a step-by-step tutorial on how to use some of the tools and databases available on these websites to make it easier to follow along. I have taken screenshots of each step, which I'll be sharing with you throughout the video. And additionally, I'll be demonstrating how to use the gene database, like which is one of the databases available in the NCBI. However, I know that there's a lot more to explore in the bioinformatics. I would really like to create more content on this topic, but if you're only interested in learning more, right? So please let me know in the comments below that some of the potential topics I could cover in future video would be uh, you know like how to perform blast searches, how to do multiple sequence alignment, or how to create phylogenetic trees and perform phylogenetic analysis, and many more. Particularly, I didn't want it to make this video too lengthy, but I'm committed also to helping you learn and understand bioinformatics. So let me know what you're interested in, and I will do my best to create helpful content for you people. Okay, so yeah, let's get into this video now. Before exploring the websites, I want to set a context uh, about giving a brief idea what bioinformatics is for people watching this video but is not from the background um, related to bioinfo anywhere. So yeah, anyway, coming to the point, what bioinformatics is. So, I can say it's an interdisciplinary field that combines computer science, mathematics, and biology to analyze and interpret any biological data given, right? It involves the use of computational tools and methods to understand the structure, function, and evolution of the biological molecules, for example, DNA, RNA, and proteins. It plays really crucial role in modern biology these days, and it enables researchers to make sense of the vast amounts of data that is generated by high throughput sequencing technologies and even other experimental methods. When I have to say what NCBI and EMBL is, so let me explain you one by one. So NCBI stands for National Center for Biotechnology Information, and it is a part of the National Library of Medicine, which is located in Maryland, and it was established in somewhere around 1988 and as a national resource for molecular biology information. And this website provides access to um, wide range of databases, tools, and resources, including PubMed, which we often use to access research papers. Then there is GenBank and BLAST, and these resources, support research in genomics, proteomics, and other areas of mol bio. When I have to talk about EMBL, so EMBL stands for European Molecular Biology Laboratory. It is an intergovernmental organization that operates from its headquarters in Germany, and it was established in 1974, similarly to promote molecular biology research in Europe. The EMBL website provides access to a range of databases, tools, and resources, including EMBL Bank Nucleotide Sequence Database, the Uniprotein Sequence Database, and there's another, the I can recall is PFAM, which is Protein Family Database. Okay, now why this video, right? Um, so I'm creating this video to provide you a step-by-step guide to using some of the key resources available on the NCBI and EMBL websites. By following along with this tutorial, you'll learn how to use these resources and to analyze and interpret biological data and gain a better understanding of the tools and techniques used in bioinformatics, right? That is only my purpose to create this. Now, whether you are a student, you're a researcher, or a professional, this video aims to provide a helpful introduction to the world of bioinfo and the resources available on this websites. Clear? Now, this will, what are the importance, right? What is the importance basically? So, both of them provide essential resources for researchers, students, and professionals in the fields of mol bio, biochemistry, and bioinform. And these databases and tools available on this website enable any user to analyze and interpret any data, like a biological data, to identify patterns and relationships and make any new discovery. So in this video, we'll be exploring some of the key resources which are available on this websites, including you know gene databases in NCBI particularly. So yeah, let's go. Now we'll visit the NCBI website. What you have to do is you have to go to the browser that you prefer. I have used Google Chrome, so you have to just go to your search bar in the Google Chrome or any other browser that you're using. Just type in NCBI and the similar home page will appear. You can see over here there's an option given all databases. You can click on this and explore the options which are given. As you can see, there's a popular resources, right? For example, there's a PubMed which we often use to access research papers for free. All right, there's a bookshelf, there's a PubMed Central, there's a another tool for performing, nucleotide, genome, there's a blast, there's many more. And then here on the extreme left side of the home page, you can see, what kind of list available, resource list is given here, right? For example, there's a chemical bioassays, and you can click on all resources, and then you'll come to know about what are the other resources that is available. Okay, so what you can do is, you can just go to this site. Everything is available for free. Go click on each options if you haven't already. So just click on it and read about it and explore on your own, because that will make you remember things. Okay, this practice is going to be really helpful if you do it on your own, right? You cannot just remember if you are weaving it. That's all. So moving next, as I said, that if you click on this all databases option, you'll see a set of options available. So these are the options and many more, right? Here you can see there's an option to scroll down. I'm not going to scroll down, and you can just do it on your own as well. So this is how the home page looks like of NCBI. Okay, that's all you have to do. Go visit and explore. Then moving on to how to visit to EMBL website, right? Just go to go to the preferred browser and type in the search bar that ebi.ac.uk. This is how the home page of EMBL looks like. Again, there's a lot of options available here. You can click on each of them and explore separately. I'm not going to take your time as well, because again, the video is going to be a bit lengthy otherwise. So yeah, now as you yeah, so here if you scroll down further, as you can see, there's a lot more to explore. Here I have clicked on the option called as overview. Okay, now if you click on the overview tab, there's a tools and data resources that opens up. You can see a lots of tools and lots of data resources that are available. You can click on each of them and explore simultaneously, right? After watching this video. And then here's, for example, I've already mentioned there's a cluster omega, right? So this is the tool. If you click on here, it will take you to the cluster omega's website, and this tool is used for performing multiple sequence alignment of DNA. If you want me to give you um, provide you with a video how to perform MSA of DNA or protein sequences, so I'll definitely make a video on that. So yeah, there's the cluster omega, then there is a blast for protein, there's blast for nucleotide as well. There are data resources available. There's UniProt, for example. This is used for it's a resource basically for protein sequence and functional annotation. So you can click on each of them, and separately you can learn more about them. Moving on further, as I said, okay, there's a blast for protein, blast for nucleotide, then there's other resources available to open for exploration. Here, this are the tools used for finding out similarity, in between the protein sequence databases or nucleotide sequence databases, respectively. Now, as I said that in this video, I'll be letting you know how to explore gene database on the NCBI website. So let's get into it. We'll be using a gene nucleotide database. All right. So there was an option here. If you watched previously, that it was written all databases. You have to click on that tab, and it will let you know what are the options available. So select gene. Okay, I have selected the gene database here, and in the gene database I'm particularly interested in knowing about cat gene, which codes for a catalase protein. Okay, I'm not specifying here in which organism I'm interested in. I have in general just typed in cat. Okay, and then you have to hit a search button. Once you've hit the search button, the search results opens up. Okay, now here you have to focus here that okay, there's not only one result. Okay, here's 82,152 results that have opened up. And here, since I had not written in which organism I was interested in, it has given me options. Right now my interest was in Homo sapiens, and my luck, the first result that has opened up for Homo sapiens. Okay, there's, if you're interested in the catalase cat gene, which codes for catalase protein to be present in Drosophila melanogaster, so you can click on this. Okay, and each results have a different gene ID. All right, so this one, the first one, which has a gene ID of 847, I'm interested in this one, and then I'm going to click on this, and then furthermore the descriptive results will open up. Okay, and similarly you can click on any of the given belows. Okay, uh, so here you can also see there's a description, okay, about of the organism in which it is present, and there's a location. For example, this, in the Homo sapien, it is present in the chromosome 11, and if you look in the presence of this gene in Drosophila melanogaster, it's present in chromosome 3L. Similarly, then you can further or move on. Then the search result will give you a kind of summary of the particular gene which has been searched, and the summary includes the official symbol, that was CAT, which is used for the gene, and the official name, which would be catalase. In my case, you can use any other gene you're interested in, for example BRCA1. Okay, any other gene you're interested in, you can just type in and get the results. All right, so I would just like to request you to explore on your own and practice as much as you can. Then you have a primary source data, then you'll come to know what kind of gene type, what is the function of this gene, etc. Okay, so as I told you, you'll get a set of data available. So as you can see on the extreme right of the slide of the home page, that the table of content, right? So if you click on summary, the summary tab will open. Here you can see the official symbol, name, primary source, and you can also get the summary of this gene. So it's here written here, this gene encodes for catalase protein, which is a key antioxidant enzyme in the body's defense against oxidative stress. Then there's catalase, which is a heme enzyme that is present in the peroxisome of nearly all aerobic cells. Okay, so you can read on your own what all the summary is all about, and then you can know more about the, if you're interested in genomic context, you can click over here. If you want to know about the genomic regions, the transcripts, which are the products, you can click here. If you're interested in knowing the bibliography, the variations, the phenotype, what are the HIV-1 interactions, pathways from PubChem, whatever you want the reference sequences, you can click on the option given here, and then whatever, if you're interested in finding out what are the related sequences, you can click here and explore on your own, right? Even you can download this data sets for your future references. And yeah, as I told you, if you click on the genome regions transcript, you can just scroll down and explore more. Okay, you can click on here, and you can also get the FASTA sequences, you can find out the GenBank, you can also download the graphics, etc. Whatever you require, you can find out here on this website. And yeah, that's all in this for this video. Thank you so much for watching, and if you want me to create more content, um, um, either on bioinformatics or any other topic, you can just comment down below and motivate and encourage me to keep on creating this content by subscribing, liking, and commenting on my video. Yeah, that's all. Thank you so much. Until then, take care of yourself and your family. That's all. Bye bye. Thank you.